Good afternoon. This is Pei Ping together with my partner, Professor and Dr. Carol Watson. We welcome you to join us for the NIH Bridge to AI program, Bridge Center, Skills and Workforce Development Education Lecture Series. I would like to introduce Professor Wei Wang, who's endowed professor of Leonard Kleinrock in computer science and computation medicine at the UCLA School of Medicine, as well as School of Engineering. Welcome, Dr. Wang.
Thank you, Pei. Today, it's our great honor to have Professor Sriram Sankararaman to give a lecture on data mangling and data wrangling. Professor Sankararaman is a professor of computer science, computational medicine, and human genetics at UCLA. He received his PhD from UC Berkeley and did his postdoc training at Harvard Medical School before he joined UCLA. His research interests cover broadly at the intersection of computer science, statistics, and biomedicine. Sriram has been recognized with many awards, including NSF Career Awards, Microsoft Investigator Fellows, and Sloan Fellows. In this lecture, he will survey a broad class of methods of data representation, exploratory analysis, confounder corrections, and data imputation. So without further delaying, I'm going to hand over to Sriram to start his lecture.
Thank you, Wei, for that introduction. And thank you to the organizers for this opportunity to present. I'm going to share my screen now. So today, I'm going to talk about issues that arise in data science and machine learning applications to do with data munging and wrangling. So when we think of the data science cycle, there are these common steps where when you start out trying to apply data science methods, machine learning methods to a problem, we want to first understand the goals, the domain, the prior knowledge. And then we go and collect some data. And then there is this whole process of cleaning the data, transforming it, visualizing it. And finally, we then apply our machine learning model. We then critique the model. We try to figure out if the results of the model make sense. And then we go back and either change the goals, change our data, and this process repeats till we are happy with the model that we have. So today's lecture is all about data wrangling, data munging, which is really in this middle before we actually do machine learning applications. And one of the things about data wrangling, data munging is in a lot of these applications, there is this 80-20 rule where most of the time spent in this process. So this is a process which requires careful study because that is where we often spend a lot of our efforts and they're critical for the success of these types of efforts. So in today's lecture, I'm going to talk about a few key ideas, a few key tasks associated with data wrangling and data munging. So we'll talk about data representation. We'll talk about exploratory analyses. We'll talk about the issue of confounders, and then we'll talk about missing data. So first, let's talk about data representation. So the key framework that we're going to be dealing with, so the ultimate goal is to apply supervised learning to our problem. So we have some training data, we have some input features and output labels that come from some unknown function. And we are trying to learn over a space of models to figure out a model that predicts well based on some input feature. So the key task is, how do we prepare our data before we can apply this model to a given data set? So when we think of representing features, the features might come in a few different forms. We might have continuous valued features, for example, measuring somebody's height. So in this case, the height is a positive real number, and it's measured in some natural units. So those are easy to represent. On the other hand, we might have discrete valued features, categorical features. So these come in a couple of different forms. One is these features have an order associated with them. So these are ordinal features, for example, the number of years somebody went to school. So there's a natural ordering. Or there are nominal features where there is no real ordering amongst the values of the features. For example, you might have a binary feature, for example, whether somebody smoked or not. And so here we might represent it by just 2 values for this feature. Alternately, you might have a feature which is telling you what experiment, which center an experiment was done in. So there is, again, no ordering amongst the different experimental centers. And so in this setting, we use what is called a one-hot encoding to represent the different values that this feature might take. So now, even with continuous valued features, the simplest category, there are a few important issues that we need to be aware of when we are applying ML models here. Oftentimes, ML models and learning algorithms can be sensitive to the scale of these features. So imagine you have 2 features where the range of these features can be drastically different. On the other hand, sometimes the units of the feature can matter. So whether you're measuring somebody's height in feet versus centimeters. So some machine learning models and algorithms are sensitive to these kinds of issues. So oftentimes we ensure that features are on the same scale by a process called standardization. So we make sure that each feature has mean 0 and in yet another classes of machine learning models, it's important that the features are close to some assumed distribution. So depending on the type of model that we're applying, we might also have to ensure features are close to some reference distribution, for example, a Gaussian distribution. Other interesting challenges in feature representation occur when we have categorical features which live in high dimensions. For example, we might have features where you have a large number of categories or concepts that we need to consider, like words in a collection of documents, or the medical diagnoses or procedure codes in electronic health records. We could one-hot encode these features. However, the one-hot encoding ignores the relationships amongst these categories. So recently, there's been a lot of success and a lot of interest in mapping features which are categorical from a one-hot encoding to a continuous feature space. And so these mappings are typically called embeddings. And one of the first successful applications of these embeddings occurred in the context of natural text. So this was a model called word2vec, which takes words that occur in natural text and maps it to a vector representation. So these embeddings need to be learned from data. So word2vec was learned on a corpus of natural documents. And there are different possible models that give us different word2vec embeddings. So one model is called the continuous bag of words model, where the goal is to learn a vector representation so that you can predict a given word based on the surrounding words of the document in which it occurs. An alternate model, the skip-gram model, attempts to predict the surrounding words based on a word at a given position. And all of these models are trained by gradient-based learning. What is interesting about these embeddings is they often preserve the semantic relationships amongst these words. So if you take the embedding corresponding to the word Paris and the word France, and if you look at the difference, so if you look at the vector that points from France to Paris, that has the same direction as the vector that points from Italy to Rome. So this relationship of the city to the country, or the capital of the city to the country, is preserved in these embeddings. Since then, embeddings have been applied in a variety of other domains. For example, they've been applied to medical codes that occur in electronic health records using this model called med2vec. So in this setup, again, we have these medical codes. These are represented by a binary vector. And these are then mapped into this space of embeddings using a neural network architecture. Another data type where embeddings have been extremely successful is when dealing with images. So it has been documented that when you look at convolutional neural networks that process images and make predictions, the different layers of these CNNs often capture interesting properties of these images, and they can be used as a representation. So when we are trying to learn embeddings, there are a few different strategies. So one strategy is to learn the embedding together with the model that we are interested in on the data set that is given to us. The challenge is for most data sets, most biomedical data sets, the sample sizes are too small to jointly learn both the model and the embedding. So the alternate strategy is to do something called transfer learning, where the embedding is learned on an external data set, which potentially has large sample sizes. And these embeddings are then applied to the data set of interest, and we only learn the model on this data set. So an example of this strategy is in the analysis of biomedical images to predict disease status. So typically what is done to analyze these biomedical images is to take embeddings that have been learned by training a convolutional neural network on public data sets like the ImageNet dataset, and to look at the embedding that is learned from these public data sets and to train a shallow model that predicts disease status. So this type of approach makes an assumption that the original data set and the applied data set are similar enough that these properties can be transferred over. There are also variations of these approaches where you can fine-tune these embeddings. So you start with the original embedding and you try to learn it together with the shallow classifier to improve performance. Now we'll talk a little bit about exploratory analyses. So we have our data. We'd like to understand some properties of the data. So that's the goal of exploratory analyses. And this can involve a few different end tasks. So one task is to clean the data. Oftentimes the data has corruptions and errors, and not every data point is going to satisfy the distributional assumptions needed by our machine learning model. So in data cleaning, we are trying to identify the relationships amongst the instances and identify outliers, data points which probably come from a different distribution than the bulk of the data set. Another goal of exploratory analyses is to identify which features are relevant or irrelevant for the task at hand. And so this is often a starting point for feature engineering and feature selection. Another goal could be to identify what we call confounders and batch effects. So confounders and batch effects basically to the fact that our data comes from not just a single distribution, but many possible distributions. And we'd like to understand the structure that underlies this differences in distribution across our data. And the goal is, once we have done all of this, this can guide the choice of the model that we end up using for supervised learning. So let's talk about outlier removal. So in outlier removal, we're trying to identify and discard data points whose values lie outside the distribution of values that are typical for this data set. So the simplest way to do this is by univariate analyses. So we look at each feature and we compare the distribution of data points for this feature, and we see if there are specific instances that do not follow this distribution. And so this can be done either by visual tests or by statistical hypothesis testing. Moving beyond univariate analyses, typically our data sets have large numbers of features, and so we need ways to visualize the representations of these different features and the instances. And this is where dimensionality reduction approaches play a big role. So these topics have been introduced in lecture 2, but we will approach them again from the perspective of data preparation and data wrangling. There are a few different ways of understanding dimensionality reduction approaches. So one is whether they are based on a linear model or a nonlinear model. So in the linear model, you have classic examples of principal components analyses, or PCA. And in nonlinear models, we have extensions like kernel PCA and newer models like TSNE, UMAP, autoencoders. The other distinction is whether these models are what we call generative or probabilistic generative models, which means can you use these models to simulate new types of data? And here we have models like probabilistic PCA and variational autoencoders. So to introduce principal components analyses, or PCA, so this is one of the most fundamental, widely used dimensionality reduction techniques. So the goal of PCA is to find a linear low dimensional representation of the data. So if you have data that looks like this in 2D, PCA is trying to find a line that best approximates our two-dimensional data. And the approximation here can be forms. So we might want to minimize the reconstruction error between the original points and the projected data. And it turns out that's the same as maximizing the variance of the projected data. So mathematically, what PCA is trying to do is to take each data point xn, which lives in a potentially high dimensional feature, and to approximate it by a linear combination of k vectors. And we constrain these k vectors to be perpendicular or orthogonal to each other. And these k vectors are what we call the principal components. So looking at it from the point of view of our data matrix, PCA is basically trying to approximate our original data matrix as a product of these two data matrices. And these two data matrices have low rank, which means we can visualize them effectively. So the data matrix given by the matrix W, that is the principal components of our data. And the data matrix given by the matrix Z, that contains the projection of our original data matrix, projection or the coordinates of our original data matrix onto each of the principal components. So now we'll apply this PCA for detecting outliers. So here's an example where we have some data where we know that there are outliers. It turns out if you now look at univariate statistics of this data set, so if you look at the distribution of values on one of the features, the distribution of values across the feature looks reasonable. Again, when we look at pairs of features, none of these data points look particularly problematic. However, when we now visualize them on the principal components plot, we now see outliers in this data set. So the reason why this is happening is because PCA is aggregating information across all of these features and summarizing the directions where there is maximal variation. PCA does have limitations. So as we said, PCA searches for a linear transformation, and that may not be the appropriate transformation for the data that we are interested in. So there have been extensions of PCA that allow for a nonlinear transformation. So one example is something called kernel PCA, where we essentially take our original data and map it to a different feature space, a nonlinear feature space, and we apply PCA to this transformed feature space. The challenge with kernel PCA is we need to know what this transformation is. Often we do not know the correct transformation to map our original data. Another limitation of PCA is PCA attempts to preserve global structure in the data. So it preserves the distances, the Euclidean distances amongst all of the data points, but it might lose local structure. So here's an example where PCA doesn't do the right thing, where the data is distributed according to this manifold. And now when you apply PCA, you get this picture, whereas what we would like is the data to be able to unroll this manifold. So it's ignoring the local structure that is present in this example. So this motivates alternate approaches to dimensionality reduction. So we'll talk about one popular approach called T-distributed stochastic neighbor embedding, also called TSNE. So the goal of TSNE is to learn a 2D projection of our data with the goal that the pairwise distances in this 2D projection are as similar as possible to the original space. So we're trying to learn this projection, and we define as similar as possible by this metric called the KL divergence between the pairs of distances in the original space and the pairs of distances in this two-dimensional space. And the way we learn this embedding is by doing gradient-based optimization on this KL divergence objective function. So the key metric that TSNE needs for it to be successful is this hyperparameter that we call perplexity. So perplexity can be thought of as the expected number of nearest neighbors surrounding each point. So if you make the perplexity large, we will have more and more nearest neighbors. And so t-SNE becomes more sensitive to global structure in the dataset. On the other hand, when you make perplexity small, the number of nearest neighbors shrinks and the algorithm becomes more sensitive to the immediate or the local structure. So the perplexity metric trades off local information to global information. And the way this fits into the t-SNE algorithm is essentially every pair of data points involved, we are computing a probability distribution over which data point can be the neighbor of which other data point. And that is controlled by a variance parameter. And so the perplexity hyperparameter is directly related to this variance parameter. So larger values of the variance parameter correspond to larger perplexities. So here's an example applying t-SNE to an original data set where the structure is known and the impact of this perplexity hyperparameter. So as you move from low values of perplexity to high values, we find that the algorithm is merging together data points that belong to these two clusters. So there is some intermediate value of perplexity where the performance of this algorithm is optimal in terms of separating points in these two clusters while keeping the points within each of these clusters close to each other. So back to our original example where PCA does not do the right dimensionality reduction, t-SNE does a much better job of keeping points that are close together the way they're supposed to be. Now, there are other approaches which also allow us to trade off local and global information. So one other class of approaches that's extremely popular are autoencoders. So autoencoders essentially generalize PCA into a nonlinear setting. So we take our original input and we transform it to a hidden layer. And then we transform the hidden layer back to an output space. So the information from the input to the hidden layer is what we call the encoder function. And the transformation from the hidden layer back to the output is the decoder function. So both the encoder and the decoder, they are jointly learned by minimizing a loss and by gradient-based learning. Another common exploratory analysis in biomedical data involves the use of multiple different data sets. So oftentimes we are collecting multiple types of data, for example, genetics, imaging, biomarkers, sensor-based data, and oftentimes these data sets are paired. So you might have one instance where you have two or more of these data modalities. So in the context of multimodal data, it's often desirable to find transformations of our pairs of data sets or pairs of instances that maximizes their similarity. So we'd like to understand how similar is genetics to imaging or genetics to sensor and so on. So we like to detect shared structure across pairs of data sets. So like PCA is a fundamental technique that allows us to detect structure in a single data set, we have an analogous fundamental technique for pairs of data sets, and this is called canonical correlation analysis, or CCA. So in CCA, we have pairs of data sets, and we're trying to find a linear transformation that maximizes the correlation of the projections of these two data sets. So it turns out, here's an example where methods related to CCA were applied to paired data sets containing genetics and gene expression. And now when you inspect what shared structure is being learned, we find that the majority shared structure corresponds to the ancestries of these different samples. CCA, again, is a linear approach. And again, there have been extensions to this approach. For example, there's kernel CCA and deep CCA, which account for nonlinearities. And then there is multi-CCA, which generalizes it from pairs of data sets to multiple data sets. So now we'll talk about another important problem in the cleaning and wrangling of data, which has to do with the issue of confounders. So typically, biomedical datasets involve samples that are collected from different populations. So populations can be a broad term. It can refer to the fact that there are different sexes, there can be different ancestries. The samples can be processed at different locations or times. There might be different experimental protocols or parameters, and there might be different biological factors that are influencing the samples differently. For example, cell type composition or cell cycle, depending on the type of data set. So the problem is many models assume that the data set is collected from a single homogeneous population, whereas in practice, that is not the case. And so ignoring this distinction can cause biases in our inference. So these factors are what we often term confounders. So a confounder is essentially a variable that affects both our features and our labels. And as a result of this, we might have a spurious association between the features and the labels that can be detected if we ignore the presence of this confounder. So here's an example from the analysis of methylation data. So the confounder here is the cell type composition. So this can change across different individuals. So in this study, an association is computed between each region of the genome where there is changes in methylation and the output, which is age. And the cell type composition has been ignored in this analysis. So as a result, what we find is there are many regions in the genome that appear to be associated with the outcome, in this case, age, whereas these are likely false positives. And in fact, for genomic data sets, it's often the case that the dominant factors affecting the variation in these data sets have to do with these confounders. For example, if you're studying microbiome data, you might have the study or the batch that you're trying to get the data from. In the context of methylation, we talked about cell type composition being a big predictor of variation. In the context of single-cell RNA-seq data, it could be the experimental protocol. And in the context of genotype data, it could be the ancestry of the individual. So a major issue in dealing with confounders is being able to correct for them. And whether we can correct for them depends on the nature of the confounder. So you might have measured confounders. For example, in the context of gene expression and microbiome data, we might keep track of the batches, which is one possible confounder. We might keep track of experimental data, like the read depth that was used for sequencing. So when you have measured confounders, supervised learning approaches are often the dominant ways to correct for them. In other settings, we have unmeasured confounders. When analyzing genetic data, the ancestry of the individuals is often unknown. Likewise, methylation data, except a few settings, the cell type compositions are not measured. So in these settings, unsupervised learning approaches are the most effective. So now let's talk about measured confounders. So one of the most common measured confounder is this notion of patch effect. So depending on when the study was performed or where it was performed, the data distribution can change. So the simplest way to deal with patch effect is to fit a linear model that accounts for these confounders. So this is implemented in these popular approaches for batch effect correction, lemma and combat. So if you're looking at the gene expression of a particular gene J in some individual I, and you're trying to correlate this to some outcome of interest, previously you would only look at gene expression in relation to the outcome of interest. But now you also add into this linear model a term that accounts for the batch information. So this model can then be fit, and there are many different approaches for fitting these models. But of course, we can also go beyond these linear models. So a common approach that goes beyond this is to use what are called mixed effects models. And more recently, there have been attempts to approach this using nonlinear models, such as deep learning. Another way to treat confounders when they're measured is to transform the data so that the batches become as similar to each other as possible. So the goal is to find shared structure or to align the data across these different batches. So we briefly discussed a technique for doing this. when you have pairs of data sets. So this is CCA or canonical correlation analyses and the extensions of this approach. So here's an example applying this technique in the context of single-cell RNA-seq data. So we have these two data sets, batches of single-cell RNA-seq experiments. And after applying CCA, we find that the cell types across these different experiments are aligned to each other. Then there is additional variants of this, for example, trying to figure out which cell types across these different batches are most similar to each other. And finally, you correct for the gene expression by aligning these different cell types. So this approach was applied to the single-cell RNA-seq dataset, where when you initially project it, you find that there are clusters that are based on the experimental protocol. And after you apply this correction that involves CCA, we find that essentially you have a well-mixed dataset where the experimental protocol no longer separates out as the dominant axis of variation. When you have unmeasured confounders, the problem of confounder correction becomes more challenging. We have to make assumptions about the nature of the confounders. So a typical assumption that is made is that the confounders have low dimensional structure, which means we can use dimensionality reduction techniques to identify these confounders. So there are different possible dimensionality reduction techniques that are used. All of these have found success depending on the domain and the type of data that is being used. So we can start with PCA. There are sparse versions of PCA because it may be the case that not all of the features are important for the dimensionality reduction. There are nonlinear versions based on Gaussian process, latent variable models, and nonlinear dimensionality reduction. So all of these techniques are potentially useful. However, in certain domains, some of these techniques tend to be used more commonly than others, probably because of a match between the assumptions underlying these techniques and the data and the type of confounders that exist there. For example, PCA tends to be used widely in getting the ancestry effects out of genotype data. Sparse PCA seems to be particularly effective in getting at cell type composition in methylation data. And the Gaussian process latent variable models and nonlinear dimensionality reduction methods have been very effective in the analysis of single-cell RNA sequencing data. But the success of all of these approaches depends on the appropriateness of the assumptions for that domain and that data set. The final aspect in data preparation has to do with this issue of missing data. So a typical biomedical data set suffers from the problem that many of the variables of interest have missing measurements. So this is an example from this data set, the UK Biobank, when we look at different phenotypes that are measured in this data set. And it turns out most of the variables are missing in most of the observations. So the data set that we are interested in analyzing looks like this, where you have missingness throughout the data matrix. So to deal with missing data, you can try to use a naive approach where you only focus on those instances where there is no missingness. So this is often termed complete case analysis or list-wise deletion. The challenge with this approach is it can reduce sample sizes drastically. And in fact, there are data sets where it may be the case that there is no sample where there is no missingness. So then this leads to approaches which try to deal with missing data by filling in the missing entries. So these are what we call imputation approaches. So the goal is to go from a data set like this, where there's missingness, to being able to fill in the missing entries. So the simplest approach to imputation is to look at each feature in isolation and to try to impute or fill in that feature. So a natural way to do this is to look at some statistic like the mean or the median value of this feature across the observed entries and to impute using the mean or the median. So this is a very simple, straightforward approach. However, it comes with certain drawbacks. It's underestimating the uncertainty in these missing entries, and it's ignoring sources of information. For example, that there are other features that could potentially be used to inform the imputation. So here's an example of applying mean imputation in the context of a data set which has missingness. And what we see is when you compare the distribution of the originally observed entries to the imputed entries, so that's the blue versus the red, there is a drastic underestimation of the uncertainty at the imputed entries, and that changes the characteristics of this data set. Next way to try to approach imputation is to try to predict the missing entries from the other entries which may potentially be observed. So in this case, we might want to predict the missing entry in feature 2 from observed entries in features 1, 3, and 4. So this is a supervised learning problem. So we are trying to do imputation by supervised learning. But the challenge here is some of the predictors themselves might be missing. So in one row, we have observed values for all the predictors. But if we go to the next row, we have predictors that are themselves missing. So we need to figure out how we're going to handle missingness in the context of these predictors. So naively, again, filling in missing values to these predictors leads to the same issue as before, where if you now do this supervised learning, you tend to underestimate the certainty in the context of these imputed entries. So to really think about imputation carefully, we need to understand the mechanism that leads to missing data. So a model to think of this is where we have our original completely observed data matrix X. We then have a matrix which is called the mask matrix. So this is a matrix where 0 means that particular entry is missing and 1 means that particular entry is observed. So given the original data matrix X and the mask matrix, we have the observed matrix, which is obtained by simply masking out the corresponding entries in X. So the goal of imputation is, given this mask matrix, we'd like to compute the posterior probability of the original data matrix X. But to do this, we really have to understand how data became missing. So we need to understand the mechanism underlying missing data. So there are a few different types of missingness, and this determines our ability to actually effectively impute missing data. So the first type of missingness is what we call missingness completely at random. So this is a mechanism where the probability that an entry is missing is completely unrelated to the data. The next one is something called missingness at random, where the probability that an entry is missing depends only on the data that is observed. So for example, you might have a population where statins are given to older people. And now when you have a young person, their statin entry might be missing. But given their age, you can then figure out whether this entry is missing or not. And finally, we have missingness not at random, where the probability that an entry is missing can depend on the observed and the unobserved data. So for example, you might have a study where you're trying to measure depression status, and individuals who are depressed might be less likely to participate in the study. So again, this is a setting where missingness might be affected by what you're trying to measure. So as we go from missingness completely at random to missingness at random to missingness not at random, these missingness mechanisms become more realistic. They also become more challenging to deal with in terms of imputation. So the key idea to be able to effectively impute data sets is to account for the fact that there is uncertainty in the missingness that we are observing. So this leads to what we call multiple imputation, where given incomplete data, we don't produce one single imputed data. We actually produce several copies of imputed data. So this accounts for the uncertainty in the imputation. We then take each of these imputed data sets, we analyze them separately, and then we pool the result to have a final analysis. So now let's see how this approach would work. So we are given a data matrix that has missing entries. So we would start by filling in this data matrix by some approach, could be either mean imputation or could be randomly filled. And then we repeat this process till convergence, where we look at each feature that we want to impute. We build a model of this feature using the current values for all the other features and only the observed entries of this feature. So this is a supervised learning problem. Everything is observed, and we can use any appropriate model for supervised learning here. Having learned this model, now we use this model to impute the missing entries in this feature. So this process is repeated till we converge, so till the imputation does not change much across runs of this data set. And this final matrix, data matrix, is what we're going to output as the imputed data matrix. So repeating this process, we have different imputed data matrices, and we analyze each of these data matrices, combine the results. So when we do this, we are accounting for variation both within a given imputed data set and across imputed data sets. So applying a multiple imputation strategy to this data set, we now find that the values of the missing entries that are imputed are more similar to the distribution of values of the original entries. So this is giving us some assurance that there isn't an underestimation of the uncertainties of these values. Okay, so just to conclude, in today's lecture, we've talked about some of the key issues when we're trying to prepare data. So we've talked about data representation, so issues of scaling and embedding. We've talked about the importance of exploratory analyses, so visualizing the data and reducing the dimensionality. We've talked about handling confounders, both measured and unmeasured, and handling missing data, where we have to think carefully about the mechanisms of missingness and how we can apply imputation, specifically multiple imputation, to deal with this. So going back to this, the data science cycle that we outlined. So we said this is a cycle, which means when we clean, transform, and visualize the data, we then apply our model. Then we critique this model, and then we go back. So oftentimes, although we think of data cleaning, data munging, data wrangling as a one-time process, it is also going to be informed by the application of downstream models and the results that we learn from this. So that's one thing that always needs to be kept in mind when we think about these models. All right, with that, I'll stop, and I'd be happy to take any questions. Also happy to follow up over email if there is additional questions. Thank you.
Thank you so much, Sriram. So we will have using the remaining time to answer a few questions. So for the audience, if you have questions, please submit them under the Q&A board, and we will go through that. So Alexander will actually read the question so that Sriram can answer them. So because I know you are sharing the screen, so it's not so easy to see the Q&A. So Alexander.
Should I exit my screen share or?
No, that's fine, because sometimes the question, depending on the question, you may want to flip back. So that's why we want to read the question to you. Alexander?
All right, thank you. The first question we have, the first technical question we have from Yurian. What is the goal to detect the shared structure in CCA, or how to interpret the shared structure?
Yeah, that's a great question. So the interpretation of the shared structure will really depend on the data and the problem domain that we are tackling. So there are a few possibilities. The assumption that is being made on CCA in some of these genomic analyses is the following. So the analysis is if you have two data sets, say two gene expression data sets that were taken at different times, the shared structure might correspond to the shared batch effect or the shared technical artifacts, and the biological signal is going to be unique. So when you're correcting for shared structure by running CCA, there's always the danger that if your assumption doesn't match the truth, for example, if biological signal is also shared, then these approaches might actually be removing that signal too. So there is always this trade-off when we are applying these approaches for, say, correcting for confounders, that you might accidentally be removing the signal in addition to correcting for confounders. So that's why being careful about making sure that your assumptions are validated is an important part of the model application or the design of these models.
Okay, great. Next question from Marco Barbero Mota. When MNAR, that's missing, it's not at random, I believe. How can you leverage that information to further inform your model? Also, what may be the causal implications of such patterns of missingness?
Right. Yes. So so oftentimes, so there's a very close connection between causal inference and missing data problems. So oftentimes, if you know something about the causal mechanism, so if you knew which variable were to cause which other variable, or which variable's response leads to a variable being missing, then these types of imputation algorithms can have substantially more power. So you can then start designing algorithms that might be able to handle things like MNAR, which you would not be able to do previously. It also turns out that causal inference is also facilitated as a problem of missing data. So when you think of causal inference, you give a person a treatment and another person is control. And the missingness is the fact that the same person can't get both the treatment or the control. So there's a very close connection between causality and the types of missingness that we see in the data. So the more we know about the underlying process that could have generated the data, the more realistic we can be in our imputation models. So this is where understanding the data, the prior knowledge, plays an extremely crucial role.
Okay, our next question from Ngo Tan Huan. Can you share more on multimodal data analysis? For example, what are the challenges and what are the solutions in multimodal data analysis? Thanks.
Yes. So, so essentially, we are now seeing more and more settings where you have different types of data that are being collected across the same individual. So you might have an individual who has their genotype data, genomic data like methylation, gene expression. They might have imaging, they might have sensor data, and you might have their medical records. So you have all of these different data sets collected on the same individual. And the big picture goal of all of this is to be able to, say, predict the outcome, predict the disease, using all of this data as best as possible. So that's what we would like to get out of multimodal data analyses. The challenge is these different data sets have very different characteristics. So there is heterogeneity. Some of these data sets are tabular. For example, data that collects your demographic information might be tabular data. Then you have imaging data, which has its own characteristic. You might have time series data that comes from sensors and wearables. And then you have genetic data, which has a different set of characteristics. So the question is, how do you build a model that allows all of these, that accounts for all of these characteristics? That's the big challenge. They also have very different amounts of signal. So sometimes if you try to combine these models, what you will find is almost all of the signal is coming from one source. All of the signal might be coming from imaging, and now no prediction. The genetics makes little contribution. And the question is that truly biology, or is that the case because we haven't figured out the right model? We haven't figured out how to integrate these data sets effectively enough.
Our next question from KJ Wilkins. Very nicely covered review of issues. Not sure if you saw the recent article about missingness. There's a link. And if so, would you welcome your thoughts on how best to build its approach with MCAR, MAR, MNAR taxonomy in mind? Thank you. The link forwards to a publication called Learning from Data with Structured Missingness. Nature Machine Intelligence, published.
Oh, okay. I
today.
don't see the link.
I can forward you I'm saving the Q&A so I can send you by email.
Yeah, for this question, maybe that can be answered offline because Sriram, it's not easy to see it, the article on the fly.
Mhm.
Let's move to the next.
Okay. Next question. In a transfer model, you said you learn embeddings on external data set and then learn the model on your data set. How does fine-tuning relate to this?
Right. So fine-tuning essentially takes the model where you might have, say, some component of the model that is obtained from an external data set. And then you have the rest of the model is the model that is specific to your data set. And what you then do is you further retrain this full model. So the idea is, if you think of this in the context of neural nets, you might have some layers of the neural nets with parameters or weights that were learned externally. And you might have some layers with additional parameters and weights. And now you retrain all of the weights. The idea is essentially you are starting out with an initialization, a smart initialization of the weights of your model, and then you're learning around this smart initialization. So that tends to be better than completely de novo approach, where you might start the weights with randomly guessed values.
Okay, next question from anonymous attendee. If the biological difference is smaller than the batch effects, the interested biological difference might not be able to be observed after correction. Any suggestion on the order of actions?
Yeah, that's a great question. So some of it some of it might be solvable by better data collection or experiment design. So this is not something that we covered here, but a lot of this some of this can be solved by careful design of experiments. For example, when samples are assigned to batches, by making sure that they're sufficiently randomized, these kinds of effects can be mitigated somewhat. So an important component here is careful experiment design. The other issue here is also thinking through whether the magnitudes are actually recoverable. So there might be settings where, given the magnitude of batch effects relative to the magnitude of biological effects, it may not be possible to accurately recover the biological effect from a given data set. So experimental design is key here.
Great. The next question we have by Sanmati Dugad. For a gene count data table where some data points are very low, is it okay to decide a threshold and filter gene count below that threshold? If yes, what would be the best way to decide the threshold?
Right. So this also probably depends on whether this is bulk versus single cell data. Yes, so if it's single cell data, you will have a lot of zeros in the gene count data matrix. And the question is how much of that represents true zeros versus how much of that is zeros due to dropout, so experimental artifacts. There are a few different approaches. So one approach is if you're trying to do differential gene expression analyses, these kinds of filtering strategies might be okay, but you might end up prioritizing certain types of genes. So you need to be careful about whether this is appropriate for the type of analysis that you're doing. The other possibility is sometimes you're doing more aggregate analyses. In that case, filtering genes based on a threshold might work okay. So yes, the answer to this will really depend on the downstream analysis that is being planned.
Okay. That's it for the technical questions. We have a question from Hema M. Are the recordings of previous lectures available? Thank you. It says Dr. Ping is going to answer this question.
We will be making all recordings available to our Bridge to AI community, as well as the scientific community and public at large very soon. Our platform requires technical construction as well as several approval process, including UCLA and NIH approval, and that's has just entered those approval process. So we think doesn't take much longer. Those things will be available. And if you are interested to get a peek preview on the lecture recordings, do contact us. We will try to see if we could have a couple of lectures ready for you earlier. Thank you for your interest.
Great. We have another technical question from Namunda Pandey. Can CCA be applied to integrate multi-omics data sets?
Yes, so there has been some recent work trying to apply CCA to multi-omics data. There are some challenges with doing that because it really depends on, so you can imagine, let's say you have 3 data sets instead of 2. It may be the case that 2 of those modalities might be closer to each other than the third one. So the question is, how do you take that into account when you do CCA on all of these together? But yes, there have been some attempts. It's very much a research question. So.
I think we answered all the questions. Thank you, Sriram, for this excellent lecture. I also learned a lot myself by watching it. And this will conclude our lecture today. And next week, we will have another lecture on statistical learning by Dr. Jessica Lee.
Sriram, that was fantastic. This is Carol Watson. That was phenomenal.